Thank you. I'm really pleased to be here. And as you say, the whole notion and progress we can make through academic collaborations nationally and internationally. So as you've mentioned, I'm a professor in women's cancers at the Institute of Cancer Research and the Royal Marsden. I also lead the BRC theme lead for cancer treatment effects. And I'd like to mention also both your next speaker, Professor Fotopoulou, and I are co-chairs of the British Gynae Cancer Society Research Group, and I'm currently the president of the Royal Society of Medicine Oncology section. So, if I can move my slides, yeah. So we know that there have been excellent progress in our treatments over the decades, more recently in the last decade, for women with advanced ovarian cancer. From chemotherapy to adding in bevacizumab, which was the first targeted therapy in ovarian cancer. But despite this, the majority of women with advanced ovarian cancer experienced disease relapse within 3 years of first-line treatment. And what's happened is that with surgery followed by chemotherapy, at some point the cancer may become more active, and it's about treating recurrence. There has been significant progress, but we can do a lot more. And together through research, we will be improving the number of women in remission and also hopefully more people cured. So I've been given about 15 minutes to talk about the ovarian cancer research program that I lead here. So I'm going to actually start with the main take-home messages. I really want you to see, and I've given some examples to illustrate the opportunities for collaborative research, going from preclinical through to clinical phase 1 to 3, translational research, and real-world experience, patient experience. I also want to highlight opportunities for training, for example, MDs and PhDs, and I hope I can show you that it's possible to be an international leader in the field, even without a laboratory, which is my case. So, starting with treatments and pioneering research, our institution, the ICR and the Royal Marsden, pioneered PARP inhibitor research with the preclinical and early phase development of the PARP inhibitor olaparib. And I was actually a PhD student at that point in the middle of my registrar training back in 2005, and actually probably didn't recognize the significance of the Ashworth Lab data with regards to targeting DNA repair defects in BRCA mutated ovarian cancer. And now here we are when this is standard of care across the world. And then Professor De Bono's drug development team showed the benefit of PARP inhibitors in BRCA mutated cancers, including ovarian cancer. And then I was pleased to be part of the team during my research to be reviewing and explaining how these PARP inhibitors actually work. But then going on from what I've just shown you, I wanted to highlight the first phase 3 clinical trial of olaparib, the same drug we saw in the laboratories in BRCA mutated recurrent ovarian cancer, where we saw a substantial benefit in delaying time before cancer progression here, a 70% improvement compared to standard of care, which was placebo. And olaparib wasn't the only PARP inhibitor. We were involved in altogether 3 phase 3 randomized trials in this setting in BRCA mutated and then non-BRCA mutated, as you can see here with the PARP inhibitor rucaparib and also niraparib. And it really shows how, as a team with research nurses, clinical fellows, sub-investigators, data teams, how the clinical trials actually make a difference thanks to the recruitment of patients in clinical trials. And then more recently, these drugs being taken to the first-line setting. And this was the SOLO-1 study of the same drug, olaparib, where after surgery, where cancer was resected and then chemotherapy and patients responded, there was, again, a 70% improvement in time before cancer worsened. And we published this work in the New England Journal of Medicine. And what was really good is to see the longer term follow-up. And I was privileged to lead the 5-year follow-up work, which we published in Lancet Oncology. And as you can see, at 5 years, the median time before progression, so the progression-free survival, was 56 months compared to 13 months with placebo. And what was really interesting was that these drugs stopped after 2 years. And so that benefit continues beyond stopping the drug. And more recently, the 7-year follow up. And here you see the overall survival. But there are many unanswered questions that we need to address as a community, not only in PARP inhibitors, but other DNA damage response inhibitors. How do we address resistance? Are combinations better? Which ones? Alan Ashworth's lab, when he was at the ICR, published in 2008 about a resistance mechanism for BRCA reversions of the mutations. Are combinations better? Can other synthetic lethal relationships be exploited? And can other patient groups with gynae cancers benefit from DNA damage response inhibitors? So, on that note, I'm going to mention a few current trials. This is an academic sponsored study through the ICR addressing PARP inhibitor resistance through the Soprano trial. And here we're actually looking at a very niche population. Patients that have had cancer worsening on a PARP inhibitor. The disease is thought to be amenable to stereotactic radiotherapy for oligometastatic or oligoprogression. And this study is underway now. As you can see, it's open across the country, or opening across the country. And I very much hope we can address the question if we can spare women from chemotherapy immediately and actually either give radiotherapy alone in selected cases or in this randomized trial, radiotherapy followed by PARP inhibitor rechallenge, in this case, niraparib. So now I'm going to move on to my other question was, can we actually use PARP inhibitors and other DNA damage repair inhibitors beyond the BRCA mutated and high-grade serous population? And this is preclinical work, almost 10 years old now, that showed that a mutation called ARID1A, or a mutation in the gene ARID1A, preclinically, they were more sensitive to small molecule ATR inhibitors. And ARID1A mutated cells seem to be more sensitive to PARP inhibitors. So, what about gynecological cancers? Well, those mutations are relevant to clear cell carcinomas and a number of others listed here. In particular, for clear cell, we know the prognosis is not as good as we'd like it to be, and certainly the responses to chemotherapy are very low. So, there's an urgent need for better treatments. So my PhD fellow at the time, who's a consultant now, Saira Khalik, part of her work was to optimize ARID1A immunohistochemistry rather than looking at mutation status, because in clinical practice, it's quicker to get immunohistochemistry and cheaper than gene mutations. So we wanted to do that piece of work so we could roll this out in this clinical trial. And I really want to showcase this study, ATARI, because it is an example of true collaborative teamwork, from preclinical lab work at the ICR, going through the Royal Marsden Gynae unit, clinical expertise and leadership, the trials unit, AstraZeneca. This is an academic sponsored study, but with scientific research externally sponsored. Translational work across multitude of laboratories, and of course centres across the UK, and actually went internationally in France and Canada, and of course charity funding here, the Lady Garden Foundation. So this trial, as a reflection of what I've just said, was actually chosen as one of the IJ International Journal of Gynae Cancer Awards a couple of years ago, and really this trial looks at whether ARID1A defective loss in clear cell carcinoma is sensitive to ATR inhibition. And for the ARID1A no loss, could we sensitize by using a combination of ATR and PARP inhibitors? So here was the trial design. So in clear cell, we had an ARID1A loss group or no loss group and gave monotherapy or combination therapy accordingly. And we also had another cohort because there are many subtypes of ovarian cancer and endometrial cancer and cervical cancer that at that point there weren't specific trials, yet this could potentially have some benefit. So we had this cohort there. As you can see, we needed to have 2 responses to go ahead out of 10. And then for stage 2, we needed 6 or more to be deemed as meeting the primary endpoint. I'm just going to show some of the headline results. What I'm showing you here is a waterfall plot. So what you see is the sum of the lesions, the target lesions, and below 0 is shrinkage, and above 0 is cancer worsening. And then on the other graph, you see time on treatment. To put it into context, on average for clear cell carcinoma, sadly, for many women, cancer can worsen within 4 to 8 weeks. So having some patients, as you can see, having a longer time on treatment without progression is of benefit. But overall, the response rate was 14%. The median duration of response was 24 weeks. So this didn't meet the primary endpoint, although, as you can see, there are some patients that clearly benefited more than others and did have substantial shrinkage for durable time. Then in the combination group, you see the results here. So very similar results. Some patients benefit more. Why do they benefit more than others? We don't know at this point. And then the final non-clear cell subtypes, so carcinosarcomas, cervical cancer, endometrioid, endometrial or ovarian cancers. We had a mix of patients here, but as you can see from the different colors, there are patients, for example, with carcinosarcoma that benefited in terms of partial responses for a durable amount of time. Altogether, we had 21% confirmed response rate. And I think this is very interesting. And at the moment, we're doing the molecular analysis to try and identify which patients may benefit more than others. I also want to highlight work related to this from my consultant colleague, James Stewart, who was my PhD fellow at the time and an NIHR academic clinical fellow. This work is under review at the moment, but he has reported with the team that PPPTR2R1A mutations may be a marker for ATR inhibitor sensitivity. And we'll be looking at this more closely and reporting that within the ATARI trial. So beyond, I appreciate this is endometrial cancer, but I think relevant to this audience, that we are now looking at this drug, ceralasertib, this ATR inhibitor, perhaps combining with immunotherapy to overcome immunotherapy resistance in endometrial cancer. And this study is underway, and it's based on results in other cancers that have shown that a combination can help overcome immunotherapy resistance. So now I'm going to move on to platinum resistance, and as an example of a collaboration, and this time with the ICR Marsden, Glasgow and Imperial. And this was the Octopus study, which was based on preclinical work and a phase 1 academic sponsored study that looked at a dual TORC1, TORC2 inhibitor in combination with paclitaxel in high-grade serous ovarian cancer. The phase 1 study was very encouraging in ovarian cancer with response rates of 52% and a median progression-free survival of nearly 6 months. So this was the Octopus study. And as I said, a true example of UK collaboration across many centres. This was the first randomized trial to assess this combination in platinum-resistant ovarian cancer. It didn't meet the primary endpoint. We published this in JAMA Oncology last year. But through work with Ian McNeish's laboratory and Uday Banerjee's laboratory at the ICR and Imperial, we did show that perhaps there's a biomarker with low tumor cell PTEN expression associated with progression-free survival in the combination arm. And this needs to be investigated further. I'm now going to move into another example, low-grade serous ovarian cancer. And this is a rare cancer where response rates to chemotherapy are far too low. With MEK inhibitors such as trametinib, the response rate is higher at 26%, but 36% of women came off this drug in the phase randomized trial due to side effects. Side effects are arguably equally important as efficacy. So we looked at 2 novel drugs, avetimatinib and defactinib. I'm not going to go through the mechanisms in detail here, but there are a number of webinars where I talk about this. I want to illustrate that this trial was based on an academic-sponsored phase 1 trial from the ICR in Marsden. Very encouraging results in this rare cancer with response rates of 46%, and it seems that toxicities appeared better. So we then went on to a global FDA registration study, a phase 2 study, to try and optimize this combination and look at monotherapy versus combination to see if 2 drugs are better than 1. And we presented the final results of the phase 2 a month ago at the International Journal of Gynae Cancer Society. And what we showed is that overall, in the KRAS mutated group of patients, responses were seen in 44% of women. And we did see responses in KRAS wild type, 17%, which is higher than what we see with chemotherapy or endocrine therapy. But beyond formal responses, as you can see from this waterfall plot, 82% of women had a reduction in their cancer lesions, which is helpful in this population where tumor burden is high. So now we're recruiting to the phase 3 study globally. You see these logos of international collaborations, and this is recruiting in the UK, so please consider referring women with this rare cancer to this trial. In the last few minutes, I'm going to mention antibody drug conjugates. They're across many cancers. I've highlighted gynae cancer trials here. And in particular for academic collaborations and leadership, I'm showing you targeting the folate receptor. We published a couple of years ago a small molecule looking at targeting the folate receptor CT900 in a phase 1 trial in high-grade serous ovarian cancer. But right now, it's the ADCs that are really changing practice. And this is the Mirasol phase 3 study that we published in the New England Journal of Medicine last year. This randomized phase 3 study showed that this is the first novel treatment to show an overall survival improvement in women with platinum-resistant ovarian cancer in a phase 3 study. It's the first ADC in ovarian cancer with proven efficacy, first folate receptor ADC, and the only FDA and EMA approved biomarker-directed therapy for platinum-resistant ovarian cancer. We are making steps forward. We've got a long way to go, but we're now seeing overall survival improvement. I'm also mentioning a HER2-directed ADC, a smaller study that we published, but very meaningful. Small numbers, but in ovarian cancer, as you see here, in recurrent disease, pretty impressive response rates, even in lower HER2 expressors as well. And this has led to FDA approval in the HER2 high population. I'm going to end mentioning treatment side effects. This is a real-world study called the Monitor UK, looking at maintenance PARP inhibitor niraparib, the real patient experience in terms of toxicities, quality of life, and clinical outcomes. But hand in hand, we're collecting ctDNA for translational research to look at PARP inhibitor resistance mechanisms. And finally, looking at older women with ovarian cancer, the Pharaoh study, 88 women across 9 UK centres. We're looking at geriatric assessments, whether it was feasible in routine oncology clinics with protocol-led interventions. And it's a collaboration with Imperial and Professor Andrea Rockel's team looking at sarcopenia on imaging, and we look forward to that. So I've mentioned toxicities and quality of life, and I said I lead the cancer treatments effect theme. So this is what we're looking at. We want to understand the biological basis of treatment-related toxicities across multimodalities, surgery, radiotherapy, targeted therapies, immunotherapy. We very much hope we can drive improving patient outcomes this way. So I've mentioned to you my main take-home messages, and I just want to say there are lots of collaborative opportunities, many listed here. I've talked about the BGCS new research group with Christina and myself being part of that in leadership. GCIG, I lead the Rare Cancers Committee. We've mentioned the AHSC, NIHR BRCs, and of course, academic UK and international collaborations. So thank you for your attention.